All right, let's take a look at the forensics of bodily fluids. They're not necessarily blood. We will deal with blood, but it's not only blood. The first question, if you see some kind of red stain, is it blood? I mean, if it's not blood, it's not even a crime scene, or it might not be a crime scene. So we want to know whether a certain stain is blood. We have the Kastle-Meyer test. We had peroxide and phenolphthalein. That'll turn green in blood. It's not completely specific because other things, potatoes, horseradish, they'll also turn green in blood, but usually you can tell if there are potatoes or horseradish there. Well, the truth is it's being phased out. We really use luminol now. We also look at Blue Star, works pretty much the same way. We spray luminol on surfaces that may or may not contain blood, and if there's blood there in a darkened area, it will glow. It's very sensitive. And in fact, even after people clean up the blood stain, there's usually some residue, and you can see it. So you can see where a crime occurred sometime before. If you just wash it off, it still will show the blood stains there. So very often, people try to clean up after themselves after they do a crime, but the luminol will show what actually happened, and it will show all the blood and the crime. Even when it's invisible, because after it's cleaned up, you can't see it, but still, luminol will show it. So it's a very important compound. There's also the microcrystalline tests that prove that it's blood, like hemoglobin. You get some precipitating agent, usually a metal ion. It'll precipitate as a crystal, like a brown crystal or a purple crystal in a specific shape. This thing can prove it's blood. It's not as sensitive as the chemical tests like luminol, and also can get contaminated. Contamination will kind of like make the test hard to see and hard to use. But the advantage of these microcrystalline tests is that they can even be used on blood that had been dried for 20 years. So there is some use to the microcrystalline tests. In order to understand a little bit more about we're not going to go into this too much, but we're going to understand a little bit about how to identify blood. We should know a little bit about immunoassays. The idea is that animals, when something foreign is in their body, they fight it off, and they develop antigens that are specifically designed to attack this foreign agent. So what we use rabbits for this. We take human blood, we inject it into a rabbit. The rabbit tries to fight it off, so it develops antibodies that are designed to attack human blood. So we take these antibodies out of the rabbit, and we now have something that will attack specifically blood, human blood. So, and the idea is we take, let's say we have the rabbit antibodies here, we have the possible blood sample here, we put them in water, this will drift, the sample will drift this way, the antibodies will drift this way. When they contact, because this is designed to attack human blood, you will see some clumping, some color change, some reaction here, because these antibodies are designed to attack blood. So you will see that attack, that clumping, that reaction right over here in between where they meet. And if it's not blood, they'll just pass through each other. You won't see this little discoloration in between. This is a way to identify whether it's human blood or not. That's the immunoassay test. So just like we've seen about immunoassays, it's true for human blood. It's called a precipitant test because there is some kind of clumping and some sort of precipitation. Not real. Well, it's the interaction that we see when we have the antibodies and the sample when they're interacting with each other. That means the antibodies are attacking blood. Therefore, we know it's blood. Now, this is true for human blood because the rabbit is designed to attack human blood. Now, other animals, if we want to know whether it's a dog's blood instead of human blood, we can do that. We can inject dog blood into an animal. So at this point, we already have the antibodies for dogs, cats, deer, in case we need to know if it's the difference between deer blood or dog blood. It's interesting that the precipitant test is very sensitive. Even mummy's blood. They managed to get a sample of mummy's blood, and it was able to identify that this was in fact human blood. So now we come to other body fluids, namely this one, the seminal stains. We kind of know which kind of crime this involves, and we'd like to know whether the crime happened. So we'd like to have some of these, some seminal stains as evidence that something actually happened. And usually, we try to ask the witness, because the witness, the victim was the witness. They might have seen where these stains would be found. The victim might say, "Oh, it was on my left arm, my right foot, or on the carpet, or I don't know." Whatever the victim will say that these stains were found. Often the victim is very useful in enabling us to find where these seminal stains, where we can get the evidence for the seminal stains. So to prove it and that it's a seminal stain, first of all, a visual examination is pretty much useless, especially if it's on stained clothing, because how can you really tell whether it's a seminal stain or not? And certainly not if it was washed. People just can't tell. What we use is we test for acid phosphatase, which is found in seminal fluid more than any other, and also a protein called P30. We'll deal a little bit with P30 later, but let's look at the acid phosphatase. This is found in seminal fluid more than any other fluid. So we use this chemical, sodium alpha naphthyl phosphate. It's spelled a little wrong over here, and fast blue D. The technique is to take a suspected seminal fluid stain, take a wet paper towel, put it on that suspected area. The seminal fluid will sort of like soak into the wet paper towel, and then you add these chemicals to that wet paper towel. If it changes color, then we know it's acid phosphatase, and we can be pretty sure it's a seminal stain. Oh, we use this 4-methyl-lumbiphenyl phosphate. It's called MUP. Same technique. Take a wet paper towel, put it on the area. You add the MUP to it. However, in this case, it doesn't change color. You put it in a UV light, and it will fluoresce if it really was acid phosphatase. Now, other things might also cause some fluorescence, but nothing within 30 seconds. So you put the wet paper towel there, add MUP to it, and immediately hit it with this UV light, this ultraviolet light. If you see fluorescence within 30 seconds, then you know this is acid phosphatase, and therefore it must have been a seminal fluid. The best proof of seminal fluid is finding a sperm cell. And so you might be asking, well, why don't we do that? The answer is that people have vasectomies. The attacker might have had a vasectomy, in which case there will be no sperm cells in the seminal fluid. Also, some people have a low sperm count, and there's so few sperm, you won't be able to find it. Also, especially if it's dry, if it's a dried stain, these cells, they cling to cloth, and trying to get them off with a paper towel or even with a swab, they won't come off. They stick to the cloth. They won't come out. So it's hard to sample them. So you won't be able to catch these the sperm cells. Also, when it's dry, they're brittle. They disintegrate very quickly. So very often we just can't find the sperm cells, even though that would be the best way to prove that it must have been seminal fluid. There is if you actually do get this thing and you're looking for the sperm cells on a microscope slide, you can add an antibody. It makes it glow green. So if there's one sperm cell there, this will make it very easy to find, because you'll see the green, you'll immediately go to it and say, oh yeah, there it is. So we do have that, and we do want to find sperm, but to prove that it's a seminal stain. But very often we can't, so we use the acid phosphatase test. Also, the P30. Seminal fluid has a lot of P30, more than anything else, and we use what they call a protein-specific antigen, the PSA. Same kind of idea. What this is.